I quickly make my coffee, and in a minute I'll be back and I open my camera.
Okay.
And then you can start. Is that okay?
Yeah, if I see you.
Yeah.
Okay, good. Hello, can you hear me, Marx?
Yes.
Good. So we can start the section before lunch. I hope the students can also hear me. I don't know.
Yes, there is a thumbs up from Melanie.
Okay.
And from Mizu.
Okay. So in this section, I would like to introduce our current ongoing project in the lab. I think I reserved 90 minutes for this, but I don't think we need 90 minutes. And I hope we can finish a bit earlier so that you can have a nice lunch break. So I'm Shinya, a project leader in the lab. So I have been working on this project using protein binders to understand how the morphogen functions. So I introduce our project briefly. So let's start something that we are very familiar with. Okay, good. So By now, I think you all agree from Martin's and Mark's lecture that DPP is very important gene. So as you can see here in the control wing, you see nice patterned wing shape, but in the mutant, in the DPP mutant, as you can see in the right side, the wing is completely gone, the patterning is completely gone, and growth is completely affected. So from the mutant phenotype, it's very clear that the gene is important, but the questions remains how this DPP gene is important. So in other words, how the DPP act as a protein, how DPP act to control patterning and growth. So the central dogma in the field is that this DPP, since DPP is a secreted molecule, DPP is thought to act as a morphogen. So as you remember from the lecture, so DPP, the morphogen is produced in the localized source in the tissue, and from there spread in the surrounding cells and form a concentration gradient. And based on this concentration gradient, the cells recognize the different amount of the ligand, and then based on this, they differentiate into the different cell fates. So this is the central dogma in the field. And indeed, by expressing GFP tagged DPP molecule morphogen, you can you can visualize such a morphogen gradient in a living tissue. So for example, here, about 20 years ago, the two groups published the GFP DPP morphogen gradient in the wing disk. So by crossing US GFP DPP flies with the flies that have a DPP GAR4, you can overexpress the GFP tag DPP. And you can see nice, strong signal in the middle. This is the region where DPP is expressed. And outside this stripe, you see nice shallow gradient. So yeah, this is 20 years ago's result. But still, after 20 years later, we are still working on the problem. For example, how does DPP, how important this dispersal is for controlling the patterning and growth. So the naive assumption is that dispersal is important for everything. So, but to ask these questions, the question is, can we manipulate DPP dispersal? So ideal experiment would be to somehow trap this morphogen in the producing cells, and you basically block the dispersal. So you may wonder, maybe you can ask the same question using a mutant, okay? Because in the mutant, the ligand, the DPP is not there. But of course, if the ligand doesn't exist, of course everything is gone. They cannot act. They cannot activate signal and so on. So it's a bit difficult, to whether the phenotype really mimic the importance of the DPP dispersal or morphogen gradient. So we invented to ask these questions. We invented such a system called MorphoTrap. Okay, so MorphoTrap is a membrane-tethered anti-GFP nanobody to trap GFP tagged DPP. So MorphoTrap consists of anti-GFP nanobody fused to transmembrane domain of CD8 and followed by entry in the C-terminal so that you can visualize where MorphoTrap is expressed. So, but by this way, you can trap the GFP tagged DPP on the cell surface so that block the dispersal of GFP tagged DPP. So this was done by the Stefan, the former PhD student in the lab, and they nicely published it in the Nature some years ago. So the system really worked very well. So as you can see, in the control, you see nice shallow GFP DPP gradient. But when you express this morphotrap in the source, here in the middle, you see the strong accumulation of GFP DPP molecule in the stride, and thereby blocking the dispersal outside. So this is super good. Okay, and you can now start to ask the importance of dispersal. However, that technically this is very difficult because first, all DPP has to be replaced, because replaced by GFP tagged molecule, because the morphotrap can only target the GFP tagged molecule. So you have to replace the endogenous DPP with GFP tagged DPP, which is not easy. So Stefan worked a lot on this genetics, to use mutant and rescue the mutant by GFP DPP. And then on top of it, you express morphotrap. So to produce this result in D here, he spent maybe a couple of years. So it's very difficult. And so when I joined the lab, so we started to work on, so because this is a big problem, so we started to generate a platform to manipulate endogenous DPP locus. So because this is the cleanest way to ask the importance of the endogenous morphogen gradient. So we generate a system to manipulate endogenous DPP locus using a transposon line inserted into DPP locus. So of course, by now, if we start this kind of project, you probably start to use CRISPR. Of course, you heard from Marx. But when I joined the lab maybe 5 years ago, people started to, we didn't know CRISPR yet. I mean, we started to use, and it was risky to apply because nobody succeeded. But when I was discussing with a former postdoc Oz, we found a nice transposon insertion in DPP locus, which contains at P site here. And so this transposon is very special because we can, by applying sequence-specific recombinase by C31, we can insert any sequence in this transposon position. So we inserted the tagged DPP using this integrase system in this locus. Okay, so this works. So you can insert the DPP which has tag here in the endogenous DPP locus. Of course, you push the endogenous DPP exon behind this tagged exon. So you can, this is not super cool because this may have an impact on your study. So then we removed this by using FreeBase FRT system as Marx introduced and Gustavo introduced. So in the end, we can generate the clean tagged DPP. Okay, so we can replace the endogenous DPP with tagged DPP. So when I joined, we designed this scheme and we tried, and it took me about 1 year, but in the end, we succeeded to generate HA tag knock-in DPP array. So, by the way, I didn't introduce much, but this HA tag, the small tag, is very important in the field, not only in the developmental biology. So it's a small amino acid sequence, which can be recognized by antibody, and you can visualize and so on. So this is very good. And this HA tagged DPP flies are homozygous viable and without any defect as far as we see. And yeah, this is good. So it shows that HA tag doesn't affect the phenotype of DPP. So as I said, by using immunostaining, we can visualize now where DPP is expressed. And you can also visualize extracellular gradient of endogenous DPP gradient. So as Mark said, I think I hope you still remember the antibody against endogenous DPP is not so good. We don't know why, but now we have a small tag inserted DPP, so we can use this antibody, commercially available HA antibody, to visualize endogenous gradient, which is very good. Okay, but this is not the original purpose of the project. Of course, we wanted to manipulate DPP morphogen gradient, the endogenous gradient, using morphotrap. So for that, of course, we wanted to generate GFP tagged DPP at endogenous locus. Of course, we tried. Actually, I first tried GFP insertion first, but it didn't work. I mean, it worked, but the GFP, such a GFP tagged DPP flies are not homozygous viable. So obviously, GFP is not good for tagging DPP. I think you still remember the size of DPP. The mature domain is about 100 amino acid, and GFP is over 200. So it's not maybe super surprising that the GFP tag affects the function. So we still don't know why GFP tag DPP is not super good. Yeah, but from the fact that HA DPP flies are happy and viable, no phenotype, so this is a good tool, but we cannot manipulate. We cannot use morphotrap to manipulate the HA tag DPP. So I was a bit sad, but we to manipulate HA this tag DPP, we took two approaches, okay, to manipulate endogenous DPP disperser. So one is to develop HA trap like morphotrap, okay, and the other one is a DPP trap, to trap DPP directly. So the HA trap is consists of anti-HA single chain antibody fused to transmembrane domain of mouse CD8 followed by MHA. So to aim at trapping HA tag DPP on the cell surface, so very similar to morphotrap system that Stefan developed. And DPP trap is similar to HA trap and the morphotrap, but consists of a DARPin fused to transmembrane domain of CD8. So DARPin is a designed ankyrin-repeat protein, so similar to nanobody or HA or single chain antibody. It's a small protein binders, but they are not. This DARPin is not the antibody based the protein binders. So as Max briefly introduced, DARPins the Pluton lab in Zurich have a platform to screen DARPins against our protein. So we collaborated to screen the DARPin against DPP. And for HA single chain antibody, actually I collaborated with my former lab in Japan, and because they happen to have this sequence. So without collaboration, we could not generate these two systems to manipulate endogenous DPP disperser. So let's go to the detail a bit. So of course, we wanted to characterize first how if they can really trap and block disperser of DPP. So if you see, so basically the result is the same. So if you see in the control, you can see nice shallow gradient of DPP. But if you express HA trap or DPP trap in the source, in the stripe, you see strong accumulation of DPP in the source. And therefore, you see nice blockage of DPP disperser morphogen gradient outside. You see, for example, this is in the posterior compartment. In the control, you see nice gradient, but in the trapping condition, you see no, almost undetectable the DPP. And nice thing about this, the setup is that everything is at the endogenous level. So the morphogen gradient that we work on is endogenous level. And you can now ask the importance of the gradient in the physiological level. So, okay. And now, here I would like you to compare the adult wing phenotypes from these trapping systems. So here I prepare the 3 crossing tubes. So of course you don't have chance to observe these phenotypes under microscope by yourself. So here I took the images from 3 conditions and saved images in the ADAM. So after this section, you go to the ADAM and you can check the image. So the 3 tubes, 3 crosses are control. Of course, you always need control to compare your phenotype. So the control is a HA-DPP homozygous flies. Okay, so all the flies in the tube are HA-DPP homozygous. Okay, and the second one is a HA trap. So the HA trap and DPP trap are expressed using the nabbing GAL4. So this GAL4 is driving US construct in the entire wing pouch area. So you basically trap the HA, the target DPP in the entire wing pouch area. So in the HA trap tube, so from this course, of course, I picked up only the interesting stuff. But of course, if you remember from the Martin's lecture, so it has cardio here. So that you pick up non-cardio. If you pick up non-cardio flies from this cross, you get HA-DPP homozygous, the genotype where you express the HA trap and the nabbing GAL4. Okay, and DPP trap is similar, so it has cardio here. Of course, in the adult wing, you select non-cardio flies. They have the construct, the transgene that you want to have. And in this case, you don't need to have HA-DPP homozygous because anyway, this DARPing recognize endogenous DPP. So again, I mean, you can select here, it's a bit missing. So here you have actually the balancer in the DARPing construct, and this has a starboard. So you can see the starboard phenotype next to it. So it's a short hair in the back. Of course, you don't have a chance to compare, to select the phenotype by your eyes. Of course, I selected the phenotype and took the image already for you. But of course, the best is that you look the tube by yourself and then look for the phenotype. But I think this year is a bit exceptional. So now what you should do, maybe during a break, maybe 15 minutes break. So you go to ADAM and download this folder, images, 3 genotypes, and then please compare the phenotypes between 3 conditions, control and the wing phenotype expressing HA trap or a DPP trap, and describe the phenotype and try to explain, because you probably see the differences between the phenotypes and try to explain why you see such a difference. And of course, you can discuss with your colleague by chatting system. Or of course, if you come up with cool idea explanation, please, you can write me or write Marcus. As I said, this is ongoing project. We didn't publish yet. I'm supposed to publish this year. Every year I say that we have to. I want to publish, and I didn't publish.
So this year, Shinya, this year you're obliged to, not supposed.
Yes, I have to. Yeah. So you don't have chance to scoop this anymore. Let's see. So I hope so. Can you go to Adam and check these images? And if everybody can check, find images, we can take a break for maybe 15 minutes or yeah, about 15 minutes, 10 minutes, and then we come back here to discuss the phenotype and interpretation of the phenotype. So
Shinya, do you give them a 10-minute break and then 10 minutes to look at the picture, or?
I think we don't need separate break. I think we, you can just open the image and you can take coffee or you can go to bathroom. I think that you don't need to spend many time for this, I think.
So you will discuss it with them in 10 minutes from now?
I think so. Or 15 minutes, maybe 15 minutes.
Yeah, okay, 27.
Maybe 20.
37.
Yes, 37 is okay. But can anybody just make sure that can anybody tell me that you get the image or?
Yes, somebody gets
Okay, good. I think, okay, good. So, okay, then let's meet maybe 20 to 12. Okay, good. So see you later. And if you have any questions, I'm around. So please write me. Okay, have fun. Good.
Good. Yeah, Shinya.
So
I'll go offline.
Okay. Me too.
Shinya.
Still 5 minutes, no?
Yep.
Okay. Okay. Hello. Can you hear me? Is everybody back? I don't know how to check. Okay. Good. So we can start. So I hope you can check these three conditions, the phenotypes from the Adam. So I should have actually shown these slides before you you take a break, but it's okay. So I prepared actually some hints for the interpretation. Yeah, basically the first two things were what I told. So which cross mimics DPP mutant phenotype? So HA trap or DPP trap? And guess why this cross mimics the mutant phenotype? And if, so as I said, the both HA trap and DPP trap, both can trap a DPP very effectively and block DPP dispersal. If so, so why do you see the difference? So this is the interesting questions. And of course, I didn't give hint, but of course, if you see the structure, the scheme of how they trap DPP, you know, you may get some idea. So HA trap actually binds to HA tag, the small tag. Of course, the antibody binds to the tag, but not DPP. Okay, and DPP trap, so is generated by DARPing against DPP. So we screened DARPing against DPP. So this DARPing DPP trap is supposed to bind to DPP directly. So this may give you some hint. Okay. So, but anyway, we go to the images that you observed. Oh, wait. So, oh, it's empty. So, okay. Next. Yeah. So, I hope you see these three different phenotypes. Of course, the control is the wild type, the HADPP homozygous wing. So a very nice patterned wing. So HA tag doesn't affect the function. Okay. And when you express HA trap in the entire wing area, you see some defects. Okay. Yeah, maybe you saw the phenotype and you try to, you know, the mark each veins. So for example, you know, L2, L3 seems there, but L4, L5, somehow they are fused. Okay. But as you can see here, the wing developed quite nicely. So here, as I showed, the HA trap DPP through HA tag, and this parcel is strongly blocked, but you still see kind of nice wing shape. Very interesting. And on the other side, on the other hand, if DPP trap is expressed using this Nabin-Galfo in the entire wing pouch, very strong phenotype. No veins, small wing, like mutant. Wait a moment. Okay. Yeah. So clearly, the DPP trap phenotype mimics DPP mutant phenotype. Okay. So the obvious question, so why do you see this difference? So obvious question is that...
Shinya,
yes?
Maybe you should mention, I mean, if you're not a very experienced person looking at wings, you might think that this is much, much more severe than this phenotype here.
Which phenotype?
Also that the DPP null phenotype, D8 over D12, seems to be much more severe than the DPP trap.
Yes.
But of course, you used a driver that is expressed only late in wing. And maybe, so the DPP trap phenotype is identical to when you inactivate DPP with the same driver using RNAi. Say like in the paper. It's much more obvious when you show this than here. When you compare the very left and very right figure, you can say one doesn't make a wing and the other one makes clearly a, it's a small wing, but it makes a wing.
That's true. So
I think, but this is virtually identical to inactivating DPP via RNAi using the same driver. You have to use a driver. So
yeah. So of course, the mutant is different from our setup. The mutant, all the cells lack this DPP in the wing disc development. Of course, they show a bit severe phenotype, severe than this phenotype. But of course, if we use a similar setup, using Navin-GAL4 and inactivate DPP using RNAi, for example, the phenotype is more or less similar to DPP trap phenotype. And in any case, the similarity between this and this, and similarity between this and this is very different. Right? This is much similar to mutant. Good. So, okay, obvious questions. So first question that you come up, of course, is, okay, DPP trap may block DPP dispersal more efficiently. So that's why you see stronger phenotype. No? Because we don't know actually how these tools work. So when we generate these two, we just cross and then see phenotype. And then the phenotype is very different. And then we need to make sense this phenotype. Okay, of course, the first question, as I said, is, oh, DPP trap may be the strong binder and block DPP dispersal much, much stronger. So that's why you see stronger phenotype. Okay, but as I said, so the both HA trap and DPP trap strongly block DPP dispersal. So upon expression of them in the stripe, in the source, you see strong accumulation and you see the blockage of dispersal outside. So, but of course, the one possibility is that undetectable small differences may give us the big difference here. So both are probably good binder and trap DPP strongly, but maybe this blocks slightly better, and maybe that's why you see stronger phenotype. So to test this, I briefly checked the PMAD, phosphomad signaling activity in the posterior compartment, in the target tissue. So in the control, you see nice graded signaling activity. But when DPP trap is expressed, in the source, in the stripe, you still see a big weak phosphorylation of MAD activity in the target tissue. In contrast, if HA trap is expressed in the source here, you see PMAD, the phosphorylated MAD signaling activity is undetectable. So meaning that, indicating that the HA trap actually blocks DPP dispersal more effectively than DPP trap. So, okay, then the idea that DPP trap blocks DPP dispersal more effectively seems wrong. Okay, so then, but why then we see stronger phenotype in the DPP trap? Okay, so because this is a bit tricky, because if you believe this, if you interpret HA trap result, you may think that DPP dispersal is not so important. But if you think DPP trap, if you interpret DPP trap phenotype as a phenotype of blocking dispersal, you may think that dispersal is super important. So So here, of course, we don't know yet why they show the phenotype, why they show the distinct phenotype. So here you can see, so of course we don't know yet answer, but if we see how they really act to trap DPP, you may find a reason. So as I said, HA trap and DPP trap, so both are on the cell surface and trap DPP, but the way they trap is slightly different. So HA trap, of course, binds to HA tag and not DPP, So, but DPP trap is a direct binder against the DPP, So if you imagine, maybe from Marx's lecture, you still remember how DPP act as a ligand and bind to receptor, activate signal. So if the HA trap HA tag, then it's possible that DPP is still free to activate signal. Okay, and DPP trap binds to DPP, so it's not super surprising that DPP cannot bind to receptor properly. Okay. So from the structure, from this the scheme, you can start to develop the idea why you see the strong difference, especially since DARPing actually block dispersal less efficiently. So to check this, we can check whether the signaling activity is affected in both cases. So indeed, when you express HA trap in the stripe using Navier-4 and DPP trap, you see strong PMAD defect in the posterior compartment where the target cells are located. But in the anterior compartment, if you see HA trap expressing wing disc, the signal is reduced, but you still see substantial signal. In contrast, if you see the wing discs expressing DPP trap, the signal is lost, everywhere, and especially in the anterior compartment. So as I showed here, it seems that HA trap HA, but still DPP can somehow activate, bind to receptor and activate signal. But in the case of DARPing, of course, the DPP dispersal is affected, but at the same time, the signaling activity is blocked because DARPing binds to the receptor. Of course, we don't know this. This is just speculation. This is scheme. We don't know structure. So we are interested in also solving the structure, whether this is true. Okay, but maybe in the next year in the blog post, maybe we can show the structure of them and then convince you that, okay, indeed, the DARPing blocks signaling too, in a way that they block receptor ligand binding. So, yeah. Okay, so here is a summary, how these binders and mutant affect to the dispersal and also the signaling. So in all the case, mutant and HA trap expressing wing and DPP trap expressing wing, dispersal appear to be blocked. I mean, in the mutant, of course, DPP is not there, so there's no ligand that can disperse. And the HA trap, DPP trap condition, they block disperse, they bind to DPP, so the DPP cannot disperse. Maybe you see some leakage, but the dispersal is strongly blocked. But in the case of signal, you start to see the difference. So in the mutant, of course, there is no ligand, so the signal is gone too. But in the case of a HA trap, signal is blocked, but not completely. Substantial signal still remains, especially in the source where DPP is trapped, because this DPP can still find the ligand receptor and activate signal. In the case of DARPins here, so the DARPin block trap DPP directly, so that most likely it blocks DPP binding to the receptor. So this appear to cause big difference in the phenotype. Okay. So this HA trap phenotype appear to reflect more or less the loss of DPP dispersal phenotype, and DPP trap phenotype reflect the combination of the loss of dispersal and signaling. Okay, so you may think that this is easy to interpret, but you know, when I started this project, I didn't have both HA trap and DARPin. I needed to generate this, and then suddenly start to see the different phenotypes. I thought, okay, both shows similar phenotype, but they show very different phenotype. And then you are in trouble. Why? And of course, it took me some time, but of course, it's interesting to think why, you know, the phenotype is there always. So you try to explain why this, you see this difference. And now, you know, we come up with this idea that, okay, you see distinct function of dispersal and signaling. And this provides very interesting indication. So first, if you go back to the original mutant phenotype, so from the mutant phenotype, DPP mutant phenotype, you know, in the textbook view is that, oh, DPP, since DPP is a secreted molecule, so DPP, this morphogen gradient by excretion, by dispersal, controls everything, all the aspects of patterning and growth. So this is a central, this has been the central dogma in the field. But now, if you think about our new result using this protein binder, we start to develop a new idea. So it seems that if you interpret this HA trap result, so the DPP dispersal is not as important as we thought. Okay, but this is not the end of the story, you know. And this actually raises many questions. So, for example, if you see this phenotype carefully, so the anterior-posterior compartment boundary in the control is here. So this is the anterior side, and this is posterior side. In the HA trap expressing wing, the anterior side is this part, and the posterior is this side. So it's seeing that anterior compartment is rather normal when DPP dispersal is blocked. So why is it so? DPP dispersal is not important at all. Why? You know, this is completely different from, you know, what we thought before. We need to explain because DPP is expressed in the late third interlarval stage in this green region. Okay, so without DPP dispersal, how come this DPP can affect control patterning and growth outside? This is the big questions. And if you see the posterior compartment now, it's clear that the region between L4 and L5 is gone, but this lateral part appears to grow rather nicely. So why is it so? How come this part can grow, you know, without DPP dispersal, without signal? So because if you see the mutant phenotype, everything is gone. So ligand is gone, signal is gone, and you see no wing. But if you block only dispersal, of course, in the posterior compartment, there's no ligand source, so you expect that there's no signal. So, but still, this part appears to grow rather nicely in the lateral part. This is very controversial. This is very different from our current view. Okay, so everything is weird. And clearly, the also important question is that, okay, DPP dispersal is important, still important, this part in the posterior compartment, the medial region of the posterior compartment, okay? but how this gradient controls, we still don't know. So the previous concept or idea was that DPP dispersal controls everything. So we have to try to think, the importance of this gradient in the entire field. But now we have to narrow down our question here. If we want to ask the importance of dispersal, we have to ask how dispersal is important here. Okay? Because if we block dispersal, the phenotype is seen only this part. Okay, so clearly the new approach creates new questions. Although the field is old, so it's very interesting now moment to work on this fundamental and old questions, because we realize that we actually don't know, we didn't understand anything. So this is kind of take-home message, and I don't know if you actually come up with the idea, maybe, because as I said that we did not publish this yet, maybe you have a better idea to interpret these phenotypes, and maybe we can still change our manuscript and adjust and publish. So if you have some better idea, you can contact me or Max, or yeah. Otherwise, I think I have, my part is done. I don't know. Okay, so yeah, I think my part is done. Marcus?
Yep.
So they can take now lunch break, I think.
Should we let them have lunch break?
Okay, so
yes.
Yeah, maybe you can take over. So if you have any questions, please write me in the chat or send me email. Of course, you can ask also Marcus. Good. Thank you for your attention.
Good appetite.
Good appetite.
When do they have to come back?
I don't
know. 1:30, 1.5 hours.
Okay, 1:30.
Long break.
Yeah, long break.
Yeah, they have time to read the paper for Friday, if they want to. Anyhow, good appetite.
Enjoy.
Ja. So, dürfte ich Sie noch schnell was fragen?
Ja.
Und zwar gerade bezüglich der Vorträge. Vom Format her, wie die gemacht werden, ist jetzt, also wenn ich es recht verstanden habe, also quasi die Idee, dass am Freitag alle Gruppen die dann präsentieren, oder?
Ja.
Wäre es echt möglich, man würde das ein bisschen ändern und auf quasi Kleingruppen machen, dass von jeder Gruppe, von jedem Paper jemand in einer Gruppe würde zum Beispiel zusammensitzen und sich gegenseitig so würde über kleine Zoom-Meetings präsentieren, weil wir haben das schon einmal im Mikrobio, so Vorträge, und es hat in der Großgruppe einfach überhaupt nicht recht funktioniert.
Das könnte man schon machen, es ist alles möglich. Solange das jemand zulässt und Fragen stellt, muss es nicht immer alles sein.
Weil in Kleingruppen könnte man einfach mehr auch dann wirklich diskutieren miteinander, so ein bisschen mehr wie ein Journal Club oder so etwas.
Lass uns das überdenken, das ist absolut, ich habe absolut nichts dagegen. Im Prinzip, eben, ihr habt keinen Spokesman, oder? Das ist der Punkt. Im Prinzip, wenn deine Meinung von der meisten von den Leuten ist, dann let's go for it. Es ist für euch, es ist nicht für uns, in dem Sinn, oder? Könntest du das vielleicht besprechen mit den Blockkursleuten?
Ja, kann ich machen.
Und dann einen Vorschlag machen. Dann können wir vielleicht auch mehr Zeit nehmen für ein Paper, also eine Stunde für ein Paper. Oder ich weiß nicht, wie das dann machen wollen. Jede Gruppe erzählt es einig in einer kleineren Gruppe und lässt dann einig zu bei einer anderen Gruppe. Oder wie das ich weiß jetzt nicht genau, was. Da gibt es wahrscheinlich auch viele verschiedene Möglichkeiten, das zu machen. Oder?
Ja, sicher.
Let's think about it. Es ist mir auch wichtig, dass die